Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Have you ever wondered how scientists can find just one tiny faulty gene swimming in a massive sea of perfectly normal DNA? Well, the answer lies in this incredible technology called digital PCR. Today, we're going to dive into how it completely changed the game, turning molecular detection from, a good guess, into an exact science of actually counting molecules. So here's what we've got on tap. We'll start with the big counting challenge. Then we'll look at how digital PCR provides a solution. We'll get into the nitty-gritty of how it works, see where it's being used out in the real world, and finally, we'll talk about what this means for the future of precision. Let's get into it. All right, section 1, the counting challenge. And really, this is the classic needle in a haystack problem, but we're talking about it on a molecular level. Think about this for a second. Imagine you're trying to spot the very first signs of cancer from just a single blood sample. Or maybe you're searching for one lone virus particle. The thing you're looking for is there, but it's totally drowned out by millions, maybe billions of other perfectly normal DNA sequences. So how on earth do you reliably pick that one thing out? And maybe even more important, how do you count exactly how many there are? And this is really where the big change comes in. You see, traditional PCR, the older method, kind of gives you a relative estimate. It's like looking at a crowd and saying, yeah, there are more people over there than over here. It's all based on how fast a signal grows. But digital PCR, or dPCR, it doesn't estimate. It gives you an absolute count. It's like counting every single person in that crowd, one by one. It tells you the exact number of molecules you started with. And that, right there, is the game changer. Okay, so how does it pull off this incredible trick of absolute counting? Well, the solution is actually pretty elegant, and it all comes down to a simple concept: divide and conquer. The whole magic trick here boils down to one word: partitioning. See, instead of running one big reaction in one tube, you take that single sample and you split it into thousands, sometimes millions of tiny individual reactions. By doing this, you're essentially trapping individual DNA molecules, isolating them from each other. And that's the digital part of digital PCR. Once everything is partitioned, each little tiny reaction becomes a super simple yes or no question. Is our target DNA in there? That's a 1. Or is it empty? That's a 0. There's no more guesswork, no more kinda. It's just a straight-up binary, black and white answer. So yeah, it's not about watching a glowing signal get brighter over time like in the old days. No, you're literally just counting up the yes chambers and the no chambers at the very end. The fancy math that turns that simple count into a final precise number, don't worry, we'll get to that in a bit. Okay, so we've got the basic idea down. Now let's walk through the whole workflow. It breaks down really nicely into a 5-step process. We'll go through them one by one. First, you've got to prepare your sample. Then comes that all-important partitioning step we just talked about. After that, we amplify the DNA. Then we make everything light up so we can see it. And finally, we analyze it all to get our absolute number. All right, step 1, sample prep. This is actually pretty straightforward, especially if you're familiar with regular PCR. You just make a master mix, you put in your DNA or RNA sample, you add all the usual PCR stuff, primers, polymerase, all that, and then you throw in a fluorescent reporter molecule. That's the little guy that's going to light up later on if our target is there. Okay, now for the star of the show, partitioning. There are really 2 main ways to do this. First, you've got droplet digital PCR, or ddPCR. This is pretty cool. It basically mixes your sample with oil, creating thousands of tiny separate water droplets. Think of it like a vinaigrette salad dressing, but with each droplet being its own tiny laboratory. Then you have microchamber dPCR. This one uses a physical chip, like a tiny wafer, that's been etched with thousands of microscopic little wells. But honestly, whether it's droplets or wells, they both do the exact same crucial job. They isolate the DNA molecules. Next up is step 3, amplification. And for anyone who's ever done PCR, this part is going to feel very familiar. It's just standard thermal cycling, making copies of the DNA. The only real difference is the scale. Instead of one reaction in one tube, you're now running thousands and thousands of these reactions all at the same time, in parallel, inside every single one of those little droplets or wells. So once the amplification is done, we move on to the final steps, seeing the results and counting them up. This is where those fluorescent probes we added earlier do their job. They hunt down and stick to any amplified DNA, which basically makes the positive partitions light up and glow. The machine then just scans everything and does a simple count. How many partitions are glowing bright? Those are our ones. And how many are still dark? Those are our zeros. It's a beautiful, simple binary readout. Okay, so you might be thinking, great, we just count the bright spots and we're done, right? Well, almost. There's one little statistical wrinkle we need to iron out. See, just by random chance, some of those tiny partitions might have grabbed more than one DNA molecule at the very beginning. It's like throwing a handful of marbles onto a grid of boxes. Most boxes will get 0 or 1 marble, but a few are going to get 2 or 3, just by luck. This is where a little bit of math called Poisson statistics comes to the rescue. By looking at the number of partitions that are totally empty, the zeros, this formula can work backward and figure out exactly how many partitions likely had more than one molecule. It corrects for that random chance, turning our raw count into that truly precise absolute number. All right, that's the how. But what about the so what? Where is this amazing technology actually being put to use? Let's take a look at how dPCR is making a huge difference out in the real world, moving from the lab bench straight to the clinic. And wow, this is where it gets incredibly cool. Just look at this list. We're talking about oncology, using something called liquid biopsies to find tiny traces of tumor DNA just floating in a patient's blood. We're talking about detecting viruses and pathogens with unbelievable sensitivity. It's used for prenatal diagnosis without any invasive procedures. It can analyze exactly how many copies of a gene someone has, or measure gene expression with stunning precision. It's even used for things like detecting GMOs and making sure our food is safe. The impact here is it's huge. And just to be clear, this isn't some pie-in-the-sky future technology. This stuff is real, and it's being used right now. This table just shows a few of the big commercial systems on the market, from major companies like Bio-Rad and Thermo Fisher. And you can see you've got both the droplet-based systems and the microchamber or nanoplate-based ones. These are the workhorses in labs today, pushing research and diagnostics forward.
Download our microbiology study app made for researchers like you.
So let's wrap this up. What's the final verdict on dPCR? Let's take a look at the good, the bad, and where this incredible technology is headed. Okay, let's break down the pros and cons. On the advantages side, it's a home run. You get that absolute number without needing to run standards, it's ridiculously sensitive and precise, and it's way more forgiving if your sample isn't perfectly clean. But we have to be honest about the limitations too. It is more expensive, it can be a bit more complicated and take more time to run, and sometimes the range of concentrations it can measure accurately might be a little narrower than other techniques. But at the end of the day, this quote really nails it. Even with those limitations, digital PCR is just a massive leap forward. The improvement in sensitivity, in precision, in just sheer reliability, it's undeniable. It has fundamentally changed what scientists and doctors thought was possible across so many different areas. And that really brings us to the big, exciting final question. We now have a tool that lets us literally count single molecules of our own genetic code. I mean, that opens up an entirely new frontier for medicine, for research, for everything. The question isn't, can we find the needle in the haystack anymore? We know we can. The real question now is, what other needles are out there that we should start looking for? With this kind of power, what are we going to be able to detect next?
Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.